Hi everyone, my name is Akanksha Nagarkar. I am an undergraduate researcher at the Downs Lab at UMass Amherst. And today I will be presenting my research, which is titled The Generation and Characterization of a Null GABAA Receptor Alpha-4 Subunit Danio rerio line. So let's start off with what is GABA? GABA stands for gamma-aminobutyric acid, and it is the chief inhibitory neurotransmitter of the mammalian central nervous system. Here we see a structure of what GABA looks like. And because it's an inhibitory molecule, it's involved in many different neural pathologies like epilepsy, Parkinson's disease, and many more. So GABA carries out its work by binding to a GABA receptor. And we will be focusing on one of those GABA receptors, which is a GABAA receptor. So what is the GABAA receptor? The GABAA receptor is an ion channel that is embedded in the membrane of a neuronal cell, as we can see here. It is selectively permeable to chloride ions. So when a neurotransmitter like GABA binds to the receptor changes shape and chloride ions can flow through. Under physiological conditions, the chloride ions flow into the neuron, and this lowers the chance of an action potential occurring. So in this way, the receptor typically has an inhibitory effect on neurotransmission. The structure of the receptor itself is pretty interesting. We can see here that it's composed of multiple subunits, and we know that there are 19 possible ones. This is what previous research has shown. And although we get lots of combinations of subunits, the alpha subunit is consistently present at the GABA binding sites. And that's pretty significant that it's pretty much always there. Another interesting feature of the GABAA receptor is that it's highly conserved across vertebrate animal species. And that makes it very easy for us to use as we study the GABAA receptor. So we use zebrafish, and they're an excellent model organism. First of all, they develop transparently and outside of the mother, and so we can really access the development of it at all times. We also get a very large clutch size, so there's we have a lot to work with. The genome is fully sequenced, and they also have the same major organs and tissues as humans. And in fact, about 84% of genes that are associated with human disease have a zebrafish counterpart. So it makes them an excellent model for disease. So what does GABA signaling look like in zebrafish? So previous studies have shown that interfering with GABAergic signaling leads to hyperactive behavior in fish. This makes sense because GABAergic signaling often leads to inhibitory responses. If we interfere with that normal GABAergic signaling, we might expect to get hyperactive behavior. Also, previous research in the Downs lab found that there are 8 GABAA alpha subunit orthologs. They're shown here. So we're just going to be focusing on alpha-4 for my research. So yeah, let's look at the alpha-4 subunit in zebrafish. And the reason I chose to focus on this is because a previous research effort in the Downs lab showed that knocking down the alpha-4 subunit while also knocking down other subunits at the same time showed a hyperactive phenotype in the fish. Furthermore, we found that the expression of alpha-4 was seen to be in discrete cells throughout the brain, including in the hindbrain, which is a part of the brain responsible for essential functions like breathing as well as muscle control. Despite how significant alpha-4 seems to be, its phenotypic expression has not yet been characterized. So this has been some pretty interesting research to embark on. And the objectives of my research was to create a stable line of zebrafish in which alpha-4 expression was knocked down in all cells. And the point of this is because one of the best ways to understand what something is responsible for is to knock it out and to get rid of it and see what happens. The next step of my research, or the next objective, was to analyze the phenotypic effect of this knockdown. So here is a kind of getting into the process and the strategy I used. So first, I would need to use CRISPR-Cas to knock down the gene responsible for alpha-4 expression, and this gene is called GABAA4. And the resulting fish would be mosaic because CRISPR-Cas, which we'll get into a little bit, it will create a bunch of mutations, and the mutations won't be germline. They'll only be in some somatic cells, and there'll be a variety of mutations. So in order to make concrete conclusions, I would really need to knock out the alpha-4 subunit expression in all the cells. And that's kind of why I would need to do something like this, where I would perform a series of crosses until and genotype the resulting fish until I would get a generation of fish that are homozygous for the alpha-4 knockdown mutation. And this would allow me to get a stable line. This is a schematic of kind of more detailed of how I did that. So I would first start with CRISPR-Cas9 to target the GABAA4 gene and create some mutations. I would then genotype the resulting fish, identify what mutations I had, and then I would perform a series of crosses and genotyping the resulting fish until I end up with a F2 generation in which there are fish that are homozygous for the GABAA4 knockdown mutation. So one of the most significant tools I used was CRISPR-Cas9, and I used CRISPR to target the GABAA4 gene in the zebrafish and create mutations. So first, with CRISPR-Cas, you need the guide RNA paired with the Cas9 enzyme. So I first designed guide RNA primers targeted to the GABAA4 gene. And then I injected the guide RNA and Cas9 enzyme cocktail into wild-type embryos that were in the single-cell stage. So this caused double-stranded breaks. It caused mutations that really interfered with the gene. I was then able to go ahead and identify which mutation I had created, which one I wanted to focus on, and have my whole null line be. So one of the mutations I created was an 11 base pair deletion in exon 4. So after the series of crosses and genotyping, this is the mutation that the resulting line of alpha-4 knockdown fish all have. So once I had established the stable zebrafish line that was homozygous for the alpha-4 knockdown mutation, I next performed behavioral assays to analyze the effect. So one of the assays I did was touch-evoked behavior. So this involves tapping the head of zebrafish at various developmental stages with a probe and analyzing any startle responses that followed. The startle response is also known as an escape behavior, and it's when the zebrafish bends its body in a C shape before swimming away from the stimulus. So this is what a wild type looks like. Tap it on the head, and then we see it kind of swims away, just to see it again. And then here is what a mutant would look like. You can see that there's a lot of bending, kind of in a C shape, in response. So you can see kind of a little bit of abnormal locomotion there. So this whole process was done in a chamber with a high-speed video camera to film the responses, and I used a larval zebrafish tracking program called Marigold, which was developed in the Downs lab to analyze the videos and determine fish movement and acceleration at various time points. So I recorded the number of C-bends and the swim duration in fish that were homozygous for the mutation, as well as in fish that had one normal copy of the alpha-4 protein. The process of analyzing the data is still ongoing, and so these graphs were made with an N of 9. So far, there were no significant differences in the C-bends or swim durations between the two groups, and this is most likely due to the low N and the high variability among samples so far. So yeah, we can see here, this would be the fish that were homozygous for the mutation and one with a normal copy. And so as I continue to analyze and go through the data, it'll be interesting to see what we find. Another assay I did was light flash, and this involves placing zebrafish at various developmental stages into a chamber. I acclimate them to darkness, and then they're exposed to a light stimulus. The number of C-bends were also obtained from these videos. So this is what the assay looks like. You can see the light comes on, and we kind of record how they respond. So the data analysis for this is also still ongoing. So from this research, I have created a null alpha-4 line that can be used in future experiments. And it would be particularly interesting to follow up with the previous screen and to knock down alpha-4 with other subunits to see if it reveals hyperactivity. And so this line can be used to investigate that further and characterize alpha-4. As I continue to go through and analyze the data, I can make some more concrete conclusions about the role of alpha-4 in locomotion and behavior. In this grand scheme of where this research lies, it will provide some insight into the role that alpha-4 plays in GABAergic signaling. So that's it so far. Thank you all so much for listening, and this work was definitely a team effort, so I must acknowledge everyone at the Downs Lab. Thank you so much.